So, good morning. My name is Erik Kinnman. I'm the CEO of NeuroVive. Happy to be here and talk about our very exciting portfolio and our company, which is in a very interesting phase as well. I can't move this one. Oh, okay. We're a Swedish pharma company focused on mitochondria and mitochondrial medicine. And we have two projects in clinical development. I'll talk to them in a moment. One is on traumatic brain injury, and that one is in phase 2. And then we have a second program, which is for genetic mitochondrial disease, which just recently went through its first phase 1 trial. In addition, we have a rich preclinical portfolio. One of the most important projects in that one is a NASH program that is ready for out-licensing. It's in a preclinical phase. We've been around as a company since 2000, so we've been doing mitochondrial medicine research for 2 decades. We're listed on NASDAQ Stockholm Stock Exchange, and we currently have a market cap of about 36 million US. So just one step back for those who don't remember why mitochondria are important. It's the powerhouse of cells. They have 2 important functions. One is to generate the energy that cells need through energy in food together with oxygen to form the ATP that cells need. And the other important function is to protect cells and also to be very much involved in controlling cell death. So obviously when there are dysfunctional mitochondria or mitochondria get damaged in some way, it's a problem for the cells, for the organs and the function of the organs. And what we are focused on is to develop drugs that enhance the function of mitochondria and also protect mitochondria. So I will talk initially about genetic mitochondrial disease. These are diseases such as MELAS, KSS, CPO, PO, and Pearson's disease. These are both mutations and deletion syndromes. It's genetic inherited diseases, and they affect mitochondrial DNA and nuclear DNA. These diseases can affect various types of organs and give various types of phenotypes. What's very characteristic with all of these diseases is that they give chronic deterioration of the organs and the functions of the organs, and there's a reduced life expectancy in these patients. Especially when it's children, they get very severely sick, and in, for instance, Pearson's disease, the life expectancy is some 2 to 4 years. The prevalence, putting these diseases together in a group, is some 12.5 out of 100,000, which means that also on a group level, these are orphan diseases. And obviously for a small company like NeuroVive, it's attractive to be in this space, since these diseases require less documentation. It's faster to get to the market and also less costly. And the risk, inherited risk with development is also lower for these type of diseases. And once on the market, you can enjoy market exclusivity, and drug prices on average are 5 times higher for these types of diseases. There's a high unmet medical need in this field. There's only one drug approved, partially reimbursed in Europe for Leber's hereditary opticus neuropathy. That one is from Santhera. Other than that, other drug companies are developing drugs, but they are about in the same phase of development as we are. So this is just to illustrate that symptoms can come from various types of organs, especially those that consume a lot of energy. So if you have faulty mitochondria in the brain, that can lead to symptoms such as mental retardation, epilepsy, Parkinson-like symptoms in children, stroke-like symptoms also in children and adolescents. When it affects the eye, you can get blind and lose the function of moving the eye. Skeletal muscles will lead to loss of function as well. And quite common is an affection also of the gut. If it's the pancreas, these patients have diabetes. And you can also lose kidney function and have a reduced function of the heart as well. So serious symptoms. Our lead program is KL1333. This is an NAD plus modulator, which is a metabolic cofactor. And what this drug does, and we've shown this in vitro and in vivo in experimental studies before going into human, is that the drug will increase energy production and also lead to the production of new mitochondria, new healthy mitochondria. The first phase 1 study was done together with our partner in Korea, and that one showed favorable, good, positive pharmacokinetics and also safety and tolerability, where the healthy volunteers only had mild gastrointestinal side effects on the highest doses. So we're now moving into the next clinical trial, where we, in addition to having healthy volunteers, will also include patients. We have the approval for initiating this study in the UK, and we now have as a goal to start this study by the end of the year. This study and the ones following that can take us to the market at about 2023, and this is a significant commercial opportunity, obviously. Our next program, also in the genetic mitochondrial disease area, is Inv tival. This is a completely different type of mechanism. It's an alternative energy source for these patients. Most of the mutations sits in the first step of the respiratory chain, the part of the mitochondria that will transform energy in food and together with oxygen to ATP. And what it does is that it's a substrate for the next step, complex 2, and by doing that, it acts as an alternative energy source and bypasses the mutation. We have our first preclinical results in vivo models, and it looks very promising, and also from a bioavailability point of view, it looks also very promising, being highly orally bioavailable and also distributed to the target organs, including the brain as well. And we're targeting one of the most severe diseases, Leigh's disease, with our program, and expect to be in clinical development by 2020. NeuroSTAT is our phase 2 program for traumatic brain injury. In traumatic brain injury, we're talking about acute severe trauma to the head, typically car accidents, sports injuries, bicycle accidents, fall accidents. In the acute phase, there's a primary injury, which we obviously cannot do anything about. But imagine this like a secondary biological helmet. What we can do is to protect the healthy brain cells. There is no protective treatment after acute injury, more only symptomatic treatment in these cases. And what this drug does is to protect the mitochondria, and by protecting the mitochondria, also the brain cells from the secondary injury, which goes on for days following the injury up to weeks. There's some 50 million patients every year having a TBI. It's a huge cost, indirect and direct cost to society, some $365 billion US per year, and the average cost in direct health costs for patients are $300 million US. So this is the concept. What our drug NeuroSTAT does is to stabilize the mitochondria through the membrane permeability transition pore. And we have orphan drug designation for this in Europe and US. We've done a first clinical trial, which was aiming at looking at safety and pharmacokinetics. That all looks good, and the drug is taken up into the brain as expected. We also looked at some exploratory endpoints in this first trial, and that one showed an effect in line with the hypothesis in terms of protecting the brain cells from damage and also having an impact on protecting the mitochondria from the cascade of calcium ions, cytochrome C, and reactive oxidative species that are released from the damaged brain cells. We've done an experimental study also in pigs in a very standardized brain trauma model. And the MRI scans that you can see are from those. On the left side, placebo-treated animals. On the right side, the panels are with NeuroSTAT, are drug-treated. And on the top, you see the black and white spot up to the right, to the left, and that is the traumatic area. And you can hardly see anything on the treated side. And on the below images, you see the average case, and for a blinded observer, there was on a group level a 35% difference. And that translates into a possibility to affect the outcome of a trauma in terms of protecting against loss of motor function, cognitive function, speech disorder, and so on, so that a patient that would be protected in this way, having an effect at this level, would be able to go back to work without being bound to a wheelchair with intact cognitive functions. So we're looking forward to a phase 2b trial, which we are planning for 2019. And if we were successful taking this to the market, it's also a big commercial opportunity. NASH, a big challenge. There are no drugs out there. I'm sure that you've heard a lot about it. The challenge is that it's some 30% of the population in the US who have fatty liver. Somewhat less in Europe, 3 to 5% have NASH. So that's when the fatty liver gives rise to inflammation. And what is dangerous for the patients is that when this leads also to fibrosis, which can lead to cirrhosis and loss of liver function. And this is becoming the most common reason for complete loss of liver function and liver transplantation. And the other risk is liver cancer. We have a drug that protects the mitochondria in liver cells, and it also has a direct action on collagen production. So it's a direct antifibrotic drug. We've shown that in a number of experimental models. And in addition to those experimental studies, we have scaled up production, and we have seen safety and tolerability in this. And we hope that this is now enough of documentation to out-license this the first half of next year. This is our overall portfolio. So I talked about the traumatic brain injury program on the left, and then KL1333 and NV 354 under the heading of mitochondrial genetic mitochondrial disease. In addition to that, we have NVP 025 in earlier stage, yet an additional very different mechanism of action. From the same program as NV354 comes from, we have out-licensed some chemistry to US Bridge Bio, who started a new company, Fortify, with the aim of developing a local treatment for Leber's hereditary opticus neuropathy as well. And in our minds, validating also the overall program that we have. And in the green bars, we have, you see products that we aim to out-license at the preclinical stage. I talked about NV556. That one is currently under discussions with several possible partners. And then we have an earlier program, NVP 022, which is addressing the more metabolic part of NASH. and we also have a liver cancer project. These are the timelines. A lot, we hope, will be happening towards the end of this year in terms of initiating KL1333 next clinical trial. The 354, we have some initial in vivo studies. We will have additional towards the end of the year and beginning of next year. And for the early programs, MVP 025, 022, and 024, we hope to have lead candidates by the end of the year. And for NeuroSTAT, the initiation of the next clinical trial next year will be an important milestone for us. We have cash until the first quarter next year. We did a rights issue in April, and with that we have a warrants program which is running this month, which could give us an additional $4 million US. Just in summary then, we believe we have what can be life-changing medicines in a portfolio which is broad and very new to the company. We have a high-performing team that has and will continue to deliver results, and we have clearly interesting near-term value triggers and possibilities of growth. Thank you very much.